So, this is Teaming with Microbes, the Organic Gardener's Guide to the Soil Food Web, Revised Edition, by Jeff Lowenfels and Wayne Lewis. Chapter 15 Tools for Restoration and Maintenance. Now that you have an idea of what populates your soils, it is time to take whatever action is necessary to ensure your soil food webs give your plants what they need in the way of nutrients and protection. Compost, mulch, and compost tea. This is when you begin teaming with microbes and become a soil food web gardener. With most soils, your first aim will be to restore a diverse and whole soil food web. As beneficial organisms return, you will see a difference not only in your soils but in your plants as well. Some areas, lawns and beds of annuals, for example, respond very quickly. Other spots will have soil food webs that take longer to establish or alter. Much of your yard's response will have to do with previous practices. If in the past you saturated your yard with commercial pesticides, herbicides, fungicides, or salt-based chemical fertilizers, you may have to completely re-establish soil food webs. This may take a year or more. Gardeners who have been organic usually need only to tweak their established food webs, employing some new practices and intensifying others. It's simple. Compost, mulch, and compost tea, and sometimes mycorrhizal fungi, are the soil food web gardener's tools, and it takes only 3 strategies to restore the soil food web using them: applying the proper kind of compost, mulching the right way, with the right kinds of organic matter, and applying actively aerated compost teas. Once established, soil food webs can be maintained with the same strategies, either alone or in combination. Employed properly, these management tools will replace conventional fertilizing with chemicals. These tools feed the microbes that feed the plants. If you keep the microbes happy, healthy, and diverse, you will have excellent results. Compost has been used to support soil food web organisms long before anyone knew they existed. It is a proven effective growing medium. Compost can inoculate in an area with microbes to support a soil food web. Properly made compost contains the entire complement of soil food web microorganisms, fungi and bacteria, protozoa and nematodes. It is also full of organic matter, which provides living space and nutrients for the gang of microbes a compost pile contains. Finished compost never smells bad, which would be a sure sign of anaerobic microbes doing their thing. It should smell earthy and fresh, and it always has a rich dark coffee color. The only caveat is that in modern times, one has to know what was used to make the compost, as many of the chemicals we seek to avoid do not break down quickly enough in compost. Organic mulches, too, are an effective soil food web gardening tool. By organic, we mean natural material full of carbon and nitrogen, namely leaves, grass clippings, and wood chips. These provide the proper environment for the soil community's organisms and plenty of organic foods for them to live on. After all, these are what make up the compost pile. Mulch is a form of cold compost. It doesn't heat up like a compost pile, but it will decay over a long period of time. By providing different kinds of organic matter as mulch, you can establish or supplement different members of the soil food web. Ones that will provide more of the type of nitrogen preferred by the plants grown in the area. Actively aerated compost tea is a liquid easily extracted from compost. A properly made, actively aerated compost tea contains the same set of microorganisms as the compost from which it derives. The term actively aerated compost tea is used to distinguish these modern compost teas from old-fashioned teas like the ones your parents and grandparents may have made by soaking a bag of compost or manure in water for a few weeks. Actively aerated compost teas are prepared by pumping air into a mixture of compost, dechlorinated water, and microbial nutrients. Unlike old-fashioned teas, which went anaerobic, actively aerated compost teas remain aerobic, and the aerobic microorganisms are the beneficial ones. The energy from the air bubbling through the mixture strips the microbes out of the compost and into the tea. Here they grow and multiply, forming a stew of beneficial food web microbes that can be applied to soil. Aerated compost teas are easier to make and much easier to apply than compost and have much higher concentration of microbes, so you don't need nearly as much tea as you would regular compost to inoculate an area. These teas can also be sprayed on leaf surfaces, where compost will not stick. Here, the beneficial microbes in the tea outcompete pathogens for food and space. More work now, much less later. Using compost, mulch, and compost tea properly will greatly reduce the amount of work it takes to maintain your yard and gardens. There is a bit of work involved in making the conversion from chemicals to microbes, but ultimately, once you gear up and make the necessary changes, there will be less to do. The microbes will be working for you. You will need to water less because the food web animals will have improved your soil's water and air holding capacity. Let me read that again. You will need to water less because the food web animals will have improved your soil's water and air holding capacity. You won't need to fertilize because there will be proper microbial cycling of nutrients in the soil, and you will be able to ensure your plants are getting the kind of nitrogen they prefer. You will have fewer plant health problems and some effective, easy-to-use tools to make things better if things do go wrong. And if all this doesn't save you time and effort, not having to rototill or turn your garden soils ever again surely will. Best of all, there are no dangerous chemicals. Nothing leaches into the water table. When you team with microbes, there is no small print to read, and no health problems for you, your family, or your parents, or your pets. You have now heard briefly what the main soil food web tools are. Each deserves and will get its own chapter. Once you start applying all the rules using these tools, we are quite sure there will be no looking back. But first, a quick look forward. As for the future, who knows? More than a century after their discovery by Dr. Frank, mycorrhizal fungi are finally available for purchase to home gardeners who need them, at nurseries and chain stores around the world, right along with other microbial products derived from the soil food web. Several other soil food web organisms and their relationship to plants are being studied as we write this. Given advancing scientific techniques, the high degree of interest in the subject, and the human as well as monetary implications of usable bioproducts, it is certain more will be taken up, more and more often, as additional tools for restoration and maintenance. Endophytes. Endophytic fungi are already being used to make medicines for people. One Taxol is perhaps the most famous of all cancer-fighting drugs. The potential for additional life-saving drugs, particularly those with antifungal, antibiotic, and antioxidant properties, to be developed for endophytes is real. To be developed from endophytes is real. This adds a new urgency to the necessity of preserving old-growth forests, lest we inadvertently eliminate important endophytes along with the trees and web of life they support. Remember, every plant tested thus far has endophytes. An awful lot is known about the role these fungi play in the growth and health of grasses. Their symbiont partner of choice, see Chapter 5. Surely organic herbicides and pesticides with application to home lawns can't be too far off, especially as more and more communities are, thankfully, eliminating or restricting the use of chemicals. At the top of our wish list is an endophytic fungi that rids the lawn of dandelions. One genus of endophytic fungi, Trichoderma, associates with plants with plant roots. Its members are found in almost all soils, and in the above-ground tree tissue as well, and are among the easiest fungi to culture. What? One of us has seen Trichoderma harzianum, characteristically green when covered by spores, in some half-consumed cottage cheese that needed tossing. The other has grown it in compost mixed with a handful of baby oat flakes or ground uncooked oatmeal. Trichoderma species are mycoparasites. They gain nutrients by attacking other fungi. They can also live off organic material. Some trichodermas parasitize only specific types of fungi. Others are more general. At the same time, these fungi somehow enhance the growth of the host plant. One strain, T-22, can reduce the nitrogen of corn crops by 30 to 40%. Strain PTA-3701 is a nematode inhibitor. Strain T-39 lives off gray mold. Many gardeners will be happy to hear. Products containing trichodermas are already used to protect all manners of commercial crop, from lentils to tomatoes. In addition to gray mold, they are effective against Fusarium, Rhizoctonia, and a bunch of other things I totally won't be able to pronounce. Sclerodium species. There you go. Frankia. Frankia is a genus of actinomycetes, the bacteria that form long hyphae-like filaments of individual organisms. Species form root nodules on host plant and produce nitrogenase, the enzyme necessary to break the strong molecular bonds of atmospheric nitrogen so that it can be converted into ammonium. Frankias are responsible for approximately 15% of all biologically fixed nitrogen. Frankias generally associate with plants growing in soil that are deficient in nitrogen. Makes sense, given their ability to be of assistance to nitrogen-starved plants by fixing nitrogen from the air. They appear on the roots of pioneer species, those that populate disturbed areas. You'll find them in soils after a volcano finishes erupting, a glacier retreats, or sand dunes shift and grow. They are important in cooler locations, like Alaska, Canada, Russia, parts of New Zealand, and the Scandinavian countries, where there are few lagoons to fix nitrogen, and where nitrogen production in the soil is therefore low. Some species invade root hairs, which once infected develop a space for them. Others grow into a root and around a cell, which they eventually invade. This leads to the development of organs that look like roots and grow from tips as new cells are added. Inside are where the frankias fix nitrogen. So far, so far, 24 different genera of plants are known to be capable of forming a relationship with frankia, and 12 species of frankia are involved. Alders, sweet ferns, myrtles, and bayberries are among the actinorhizal plants, as they are known, because of their relationship with actinomycetes. Some studies suggest that frankia species can fix nitrogen without the association with a plant root, merely by being in the rhizosphere. This makes them a potential biofertilizer for nitrogen and opens up the possibility of gardening in nitrogen-deficient soils. We have no doubt that the nitrogen-fixing powers of frankia will soon be available to gardeners. Cyanobacteria. Cyanobacteria, another group with tool potential, have been around for at least 3.5 billion years and are found everywhere, on land and in the oceans. Though gardeners are most likely to encounter them as the slippery dark slime on the sides of clay pots or coating damp wood decks. The chloroplast, the organ that makes plant food during photosynthesis, is actually a cyanobacterium evolved in a symbiont living inside an algal partner, a combination that resulted in plants as we know them. One important cyanobacterium grows on the leaves of little aquatic ferns that float in rice paddies. It produces nitrogen for the ferns, and when the ferns die, become a great resource of nutrients for the microbes that feed the rice plants. Like other bacteria, these single-celled photosynthetic microorganisms often form colonies, sheets, and mats of long filaments. They grow in the top 10 inches, 25 centimeters of soil, absorbing water, where there is water, and leaving behind them as they migrate a sticky substance that links soil particles and helps prevent erosion. Imagine products that improve soil structure, absorb up to 10 times their weight in water, and can protect plants from drought and adverse conditions. Undoubtedly, cyanobacteria will play a part in making such products a reality. Rhizobia. First discovered in 1889, rhizobia are yet another important group of nitrogen-fixing bacteria that associate with the roots of legumes, peas, soybeans, lupines, clovers. They don't fix nitrogen on their own, but only after they enter a root, which forms a nodule in which the colonies of bacteria grow and work. In return for the nitrogen produced, the plant provides the bacteria a place to live. Carbons and proteins as nutrients and access to oxygen. I need to reread that. In return for the nitrogen produced, the plant provides the bacteria a place to live, carbons and proteins as nutrients, and access to oxygen. It's a great relationship, with some of the nitrogen produced left in the soil, available to other plants and members of the soil food web. Rhizobia are available for purchase by the home gardener and are an effective and fast-acting tool when the seeds and roots of legumes are inoculated. Root hair reaction occurs about a minute after inoculation, and rhizobia-based nitrogen is produced in about 2 weeks. This is instant teaming with microbes. Phosphorus solubilizing bacteria and fungi. Much of the phosphorus in fertilizers becomes chemically locked up in soil with calcium, magnesium, iron, and aluminum to form insoluble phosphates. In these forms, phosphorus is not available to plants. Phosphorus solubilizing bacteria and fungi, as their name reveals, produce organic acids and capable of converting insoluble phosphorus into a form that can be taken up by plants. There seems to be a relationship between endomycorrhizal fungi and phosphorus solubilizing bacteria. The phosphorus freed up by soil bacteria must be quickly absorbed, lest it converted back to an insoluble form. If endomycorrhizal fungi are nearby, some of this free phosphorus is absorbed and delivered to the host plants. The bacteria may travel with a hyphae in search of phosphates. It is even possible that the endomycorrhizal fungi stimulate the plants to make more exudates to attract more solubilizing bacteria. In one study, inoculating sugarcane with phosphorus solubilizing bacteria resulted in an increased yield of 12% and required less fertilizer, reducing costs. Since all plants depend on phosphorus for the production of energy, these bacteria are the subject of intense study. Already, products on the fringes of home horticultural market make use of them, and it's an easy bet to predict more bioproducts will feature them. Rhizobacteria. Many rhizobacteria that live in the rhizosphere produce special metabolites, a fancy word for all substances produced by metabolism, including gibberellins, plant growth regulators, widely used in the nursery and agricultural industries, and cytokinins, plant growth regulators that promote cell division and greatly aid plant health and development. These plant growth promoting rhizobacteria, or PGPRs for short, are producing enzymes including chitinases, which are necessary to break down chitin, the main component in fungal hyphae walls. PGPRs have been found to aid in nutrient uptake and even solubilization of phosphorus. In some cases, they cause increased branching and development of root hairs. What other metabolites there are remains to be seen, quite literally. What we do know is that with each discovery, a whole new frontier opens, and inevitably, more additions will be made to the soil food web toolshed. So make room. Yeah, I'm gonna pass on the GTA modded stuff there, Primo. No thanks. Yeah, we're in space reading. That was chapter 15 of Teaming with Microbes, the Organic Gardener's Guide to the Soil Food Web, revised edition by Jeff Lowenfels and Wayne Lewis. Thanks for joining us for the sesh. I'm gonna get a bong ripping before I read the next chapter. That was the shorter one of the two. Oh my goodness. Oh. So sorry. Oh. Some people have some crazy usernames, I tell you what. Oh my. Thanks for joining us for the book reading. Getting together and trying to learn more about what's going on in the soil, the microorganisms therein, their relationships directly, indirectly. Today, the big one was, to me, was learning more about the trichoderma fungi. Trichoderma harzianum is something that we have and use. It's something that is a parasitic fungi that goes after other fungi, while also boosting the health of the host plant that it's chilling with. I thought that was awesome to read. I had really been wanting to look into it more as I sponsor. I'm sponsored by Herb doing the thing, so it was really cool to check that out today. Yeah. Oh my. Gonna go ahead and finish this bong bowl and get back to the next chapter. That sounds awesome. Blue Dream Kush getting rolled. Get back to this. Sounds like Rick woke up. Morning, man.
Good morning.
How you doing?
Chris just woke me up, told me he has a box full of food.
No. Is he here?
Yeah, no, Christopher across the street.
Yeah.
I just picked it all up.
Alright. Very cool. Well, I thought it was more of the school food. Have a friend that's got a kid, and they signed up for a school program for getting food while at home, and they give us the food that they get is just crappy cafeteria food. I used to freaking work at a school for a while, so I'm just over cafeteria food. You ever been exposed to something so much you get burnt out on it? Me, cafeteria food. Sorry. Yeah. Alright, so this is chapter 16, Compost. Compost is a whole universe of diverse soil food web organisms. Never mind the huge numbers in good, fertile garden soil. The number of organisms per teaspoon in compost, especially in microbial populations, are simply too large to fully comprehend. Oh, yes. Nobody likes reheated cafeteria food, though. I have to reread that, I'm sorry. Never mind the huge numbers in good, fertile garden soil, The numbers of organisms per teaspoon in compost, especially the microbial populations, are simply too large to fully comprehend. Up to a billion bacteria, 400 to 900 feet, 150 to 300 meters of fungal hyphae, 10,000 to 50,000 protozoa, and 30 to 300 nematodes. In addition to extremely high microbial numbers, compost contains all manner of microarthropods and sometimes worms. It teems with life. Morning, sis. I hope you're well. Rule number 4. Compost can be used to inoculate beneficial microbes and life into soils around your yard and introduce, maintain, or alter the soil food web in a particular area. Establishes the use of compost as a major soil food web tool. Holy shit, what did I just read? Let me reread that. Compost can be used to inoculate beneficial microbes and life into soils around your yard and introduce, maintain, or alter the soil food web in a particular area. Rule number 4 establishes the use of compost as major soil food web tool. I'm alive. I'm sorry. I had to go hawk up something. Oh. Speck of dust in the throat. It was horrible. So sorry. That's still alive. Boy, stay hydrated. Thank you. Thanks for hanging out. Oh my goodness. That's so funny that JD enjoyed the sesh. Yeah. I live in the desert. There's dirt and dust in the air. Sometimes it gets nasty. So sorry. Oh my goodness. Alright. Rule number 5 elaborates on this. Adding compost and its soil food web to the surface of soil will inoculate the soil with the same soil food. Wait. Oh my god. I am high too, so that just was hard to read. Let me try that again. Oh my goodness. She sells seashells by the seashore. And then read this sentence. Alright. Rule number 5 elaborates on this. Adding compost and its soil food web to the surface of the soil will inoculate the soil with the same soil food web. And the organisms in the compost you apply to your gardens, trees, shrubs, and perennials will spread life as far as they can. It is microbial manifest destiny but you can best satisfy a plant's nutrient needs by adding compost with the right microbial domination. Oh my. Very cool. I am a Native American, mostly, but all sorts of things. American trash? Uh, yeah. What was it? Oh gosh. Yes, Lebanese. Part of family comes from Lebanon. Traits from Lebanon, more so. Oh my. Let me see. Oh. Let me reread this. I'm so sorry. The organisms in the compost you apply to your gardens, trees, shrubs, and perennials will spread life as far as they can. It is microbial manifest destiny, but you can best satisfy a plant's nutrient needs by adding compost with the right microbial domination. Oh, very cool. Not all compost are the same. Most gardeners don't give compost much thought. They make or buy it, and they apply it. It is all the same. There is more than one kind of compost, however, which is something that amazes many veteran compost makers. We too thought that all compost, no matter what went into it, had the same biology and pH in the end. But surely upon reflection, and especially after you know something about the soil food web organisms that make up compost, the idea that the end product is always the same doesn't make any sense. As with almost every other system, what goes into it does have something to do with what comes out at the end. The fact of the matter is that by using just a bit of soil food web science, you can make either compost that is dominated by fungi or compost that is dominated by bacteria. Excuse me. It all depends on what you put into the compost pile or bin to start. And because some plants prefer their nitrogen in ammonium form, and some in nitrate form, making compost that fosters the production of one of these forms of nitrogen over the other makes real sense. How to make compost. Yeah, there are some there are some pictures, but it's mostly diagrams right now. Yeah. Okay. So, you've got the chart here. It says bacteria populations in fertile soil and compost. It's a scale of 0 to 100. 0 at the bottom, 500. Oh, sorry, 0 to 500. 500 at the top, 0 at the bottom. The chart at the bottom is also about fungi. Alright, so top chart. Bacteria, its presence in the soil. Fungi, presence in the soil. Fertile soil, minimum. Fertile soil, maximum. And then compost is just like blown up numbers. Yeah. Yeah. Oh yeah, some really sad bar charts. You know, yeah. There's some good pictures, but not at this point in time. Yes. My sister is. Yep, I'm just a farmer on the internet. Were we reading How to Make Compost, I think was where we were. Farmers have been using compost to improve their soils at least since the time of the early Romans. It was only in the last century that compost took a back seat to chemicals when it came to growing things. Before that, if you worked on a farm or in a garden, you routinely used compost and manure to increase fertility. Oh, yes. This all changed when internal combustion engines replaced the horse. And fewer homes, particularly in urban settings, featured chickens, cows, pigs, and other livestock. Agriculture and horticulture required chemicals because there was a death of manures and thus compost. Making and using compost has made a strong comeback among home gardeners and has even become politically correct. Composting conserves valuable landfill space by recycling at least some of our household waste. Dozens of compost bins are commercially available, and a like number of books can tell you how to make compost in myriad ways. At the heart of every composting system, however, are the soil microorganisms, the members of the compost food web. They are the ones that make compost, no matter what method is employed. Their metabolic activities creates the heat and byproducts that make the composting process work. This is a chapter, not a book, on composting. What we will describe here is just a bit of the science behind composting and a few basic procedures for making compost at home. Once you have made a few batches, you can experiment and create a system that best fit your plant's needs and your climate, space availability, and even spousal demands. Besides the necessary soil microbes, composting requires heat, water, air, and organic material with the right amounts of carbon and nitrogen. All are mixed in the proper ratio. Organic materials are easy to come by. Grass clippings, autumn leaves, wood chips, straw, sawdust, branches, and virtually all kitchen scraps, except meats and fats. Human and pet feces should not be composted because of the possibility that disease organisms might survive even the high heat of the compost processes. For the same reason, we personally discourage the time-worn practice of using other manures in compost. Why take the risk when you don't know what kind of antibiotics and other drugs are used to feed the animals? Who wants to be worried about E. coli? Bacteria, fungi, and other microbes seek mostly carbon from the organic matter in the compost pile, just as they do in the soil. This fuels their metabolism. The microbes also need nitrogen to make the enzymes used in the decay process and the proteins, including their chief component, amino acids, that are necessary to build structure and enzymes. Moisture is necessary to provide the optimum environment for the microbes and to prevent them from dying or going into dormancy. You cannot have active bacteria, protozoa, or nematodes without the water necessary for their transport and other life functions. Air is needed because the beneficial soil organisms that break down carboniferous and nitrogenous materials are aerobic. They breathe air, they require oxygen. It is true that anaerobic conditions can develop in a compost pile and decay will occur under these conditions as well. However, so will the production of things detrimental to plants, such as alcohols, of which as little as 1 part per million will kill plant cells. Obviously, then, it is important to keep up compost piles. Obviously, then, it is important to keep compost piles aerobic, which is why compost piles are turned and opened up, bringing air into the system. Finally, the heat required for composting does not come from the sun, but rather from the soil life's metabolic activity, most of it from bacterial activity. As you will see, this heat is what creates an environment that increases populations and causes them to change in character at the appropriate time during the composting cycle. Mix these ingredients in the right proportions, and you will end up with a rich, crumbly, dark, coffee-colored, sweet-smelling humus soil that also happens to be full of life. Though it could take as long as a year or more, it is possible to make good compost in as little as few weeks. But no matter what method is employed, it is the microbes that do most of the work. Mesophilic and thermophilic stages. Composting material goes through 3 distinct temperature phases. The first of these phases is the mesophilic. Mesophilic organisms thrive in moderate temperatures between 68 and 104 degrees Fahrenheit, 20 and 40 degrees Celsius. Even in this first stage, work begins on the straight, difficult to digest chains of cellulose, which are broken into smaller chains of glucose. Bacteria are particularly kept at depolymerization. Oh, Jesus. I butchered that so bad. Let me reread it. Even in this first stage, work begins on the straight, difficult to digest chains of cellulose, which are broken into smaller chains of glucose. Bacteria are particularly adept at depolymerization, as this process is known. Meanwhile, brown rot fungi, Basidiomycetes, regular mushrooms, and certain bacteria are active breaking down other difficult to digest material. These microbes produce endospores, spores that are resistant to chemicals and heat. This enables them to survive the next hotter phase of composting, and they return when temperatures cool. Larger soil organisms join the fungi and bacteria, breaking apart organic matter in the pile as they search for food, and microbial activity in the guts of some of these animals results in further chemical breakdown. All this metabolic activity creates heat, raising the temperature to 104 degrees Fahrenheit, 40 degrees Celsius. At this point, it becomes too hot for the continued activity of the mesophilic organisms, and those adapted to higher temperatures take over. In case you are wondering how a compost pile heats up in the spring after a frozen winter, it is simple. Some bacteria are psychrophilic, meaning they thrive at temperatures just above freezing, though some of them, the really cool bacteria, can continue to operate at temperatures as low as 32 degrees Fahrenheit, 0 Celsius. The metabolic activity of these cool-loving bacteria increases the temperature of the pile just enough to wake up the higher temperature mesophilic organisms so they can take over. Organisms in the second stage of composting cycle, the thermophilic phase, can withstand temperatures of 104 to 150 degrees Fahrenheit, 40 to 65 Celsius, and over. During this period, the complex carbohydrates are fully broken down. Some proteins are also decomposed. Hemicelluloses, more resistant structures, are decayed. Many more bacteria and fungi join in or begin to play more prominent roles. Their metabolic heat causes the temperature in the compost pile to continue to rise. These high temperatures also kill off pathogens that might be in the mix. These first 2 stages take place very rapidly. A properly made compost pile should heat up to 135 degrees Fahrenheit, 57 Celsius, in 24 to 72 hours, typically. If you have the right mix of carbon to nitrogen, the center of the pile will heat up to 135 Fahrenheit, 57 Celsius, in a day, and 150 Fahrenheit, 65 Celsius in 3. If the pile is not heating up, then you need to turn it. That is, switch the inside and bottom materials in the pile with the outside and top materials to add oxygen. If that doesn't work, add fresh green material, and these are full of easy to digest sugars that will supply bacteria the food they need. Newspaper, fruit pulps, or commercial compost inoculums can also be added to help a pile heat up. You have to monitor compost piles. It is advisable to keep a pile between 140 Fahrenheit, 60 Celsius, and 150 Fahrenheit, 65 Celsius, for at least a few days, because at this thermophilic temperature, pathogenic microbes in the compost are killed. At 150 Fahrenheit, 65 Celsius, weed seeds are also destroyed. Never let a compost pile get over 155 Fahrenheit, as this will start to burn off carbon. To temporarily cool an overheated pile, turn it. Yes, turning encourages both heating and cooling. Not only does this open the pile up to air, it ensures all the material in the pile gets treated. If turning doesn't do the cooling trick, add water or more brown materials. Changing the ratio of green, easy to digest bacterial foods to more fungal foods. Since bacteria are the primary heat generating organisms, this will slow things. There is nothing wrong with sticking your hand in the pile to gauge the heat, or you can stick a long gutter nail or metal rebar pipe into the pile. These transmit heat and will feel warm when things are going right. A thermometer is more precise, however. You can buy a soil thermometer designed for the purpose, or use an oven thermometer. Maturation stage. As the complex proteins and carbohydrates are broken down and begin to diminish, there is a reduction of metabolic activity, and the temperature in the pile starts to decrease. The mesophilic organisms, whose specially protected spores enable them to survive the higher heat stage, reassert themselves and replace the thermophilic organisms. The compost enters the final maturation stage. During the maturation stage, the decay of the most resistant plant During the maturation stage, the decay of the most resistant plant component, lignin, is completed. The bonds holding the chains of alcohols and lignin together are extremely strong and structurally much more difficult to attack and break apart than almost anything else in the pile. The actinomycetes, the chain-like bacteria that resemble fungi, continue their attack on these really difficult to digest plant remnants. These are the same organisms that impart the earthly smell associated with good compost and soil, which comes from their decay of cellulose, lignin, chitin, and protein. The major fungal participants in the last stage, the basidiomycetes, are still at work. Jerky sales are postponed for a while. Also during this maturation stage, physical decomposers continue to support the microbial team, grazing by nematodes, springtails, centipedes, and others, because the populations of fungi and bacteria, grazing by nematodes, springtails, centipedes, and others cause the populations of fungi and bacteria to increase. And as these microbial populations increase, so do their soil binding activities. Lots of nematodes, which were killed by the heat of the thermophilic stage, but those that survive have lots and lots of bacteria and fungi to eat. As a group, they do well. Worms, too, work the organic matter in the pile, exposing it to bacteria and their coating particles with a mucus that binds them together into aggregates. Ants, snails, slugs, mites, spiders, road beetles, and sow bugs can come into pile and open up the organic matter as they forage, shredding it and making it easier for microbes to attack. The end result of all these organisms going about their day-to-day business is compost. It is best to keep the compost pile between 104 and 131 Fahrenheit, 40 and 55 Celsius, after the initial thermophilic run-up to 150 Fahrenheit, 65 Celsius. Yeah. Make sure that the outside of the pile gets turned into the center so all the material decays. If the pile drops below 104 Fahrenheit, 40 Celsius before it is mature, consider adding some more green, high in nitrogen material. If it stays above 131 Fahrenheit, 55 Celsius, consider adding more brown carbon-containing material. Of course, aerating a pile will always initially cool it down, and if you have the strength, repeated turning is the only control you need. Watering a pile down will also cool it, but this is a more drastic step. The pile needs to remain moist throughout the process. Don't let it dry out, but don't let it become so saturated that there is no air to supply in the pile. You may have to add water as you turn the pile, or cover it to keep rain from soaking it. If all goes well, and it usually does, compost happens. After 2 or 3 turns, your pile should be compost. It is finished, or mature, when you cannot reorganize what's in it. C/N ratio in fungal bacterial dominance. Fungal versus bacterial dominance. The ratio of nitrogen has to be right in order to make compost. The ideal C to N ratio for this purpose is somewhere around 25 to 1 to 30 to 1, the carbon to nitrogen ratio. If you have too much carbon, nitrogen is quickly used up and the decay process slows. If you have too much nitrogen, organisms snatch it up and the carbon is vented to the atmosphere, or mixed with water and washed out of the pile. But at the ideal ratio, things go fast and decay is complete. Often, gardeners divide available composting materials into 2 categories, brown and green. Aged, brown organic materials support fungi, while fresh, green organic materials support bacteria. Rule 6. Brown items, including autumn leaves, bark, wood chips, twigs, and branches, obtain carbon. Carbon provides members of the soil food web with energy for metabolism. Green items, such things as grass clippings, fresh-picked weeds, kitchen scraps, contain plenty of the easier-to-digest bacterial food and are good nitrogen sources. The fresher the green items, the more nitrogen it will contribute to the pile. Nitrogen provides soil food web organisms with building blocks for proteins, which are used, among other things, to produce the digestive enzymes necessary in the decay process. Not all organic waste at hand have the ideal carbon to nitrogen ratio. Sawdust, for example, is 500 to 1, and paper is 170 to 1. The two organic waste you should have uh, the two organic waste you should have a good supply of are grass clippings, 19 to 1, and tree leaves, 40 to 1 and 80 to 1, mixed together. These will give you the near proper ratio. It is possible to manipulate compost materials so that the end product is highly fungal or highly bacterial, or a balance of the two. Simply increase brown materials to increase the amount of fungi, or green materials to increase bacterial amounts. A good mix of materials for a fungal recipe is 5 to 10% alfalfa meal, 45 to 50% fresh grass clippings, and 40 to 50% brown leaves or small wood chips. A suitable bacterial recipe would include 25% alfalfa meal, 50% green grass clippings, and 25% brown leaves or bark. Again, the green materials that go into compost provide simple, easy-to-use sugars and lots of nitrogen and are great for supporting bacteria. The brown materials in compost piles consist of difficult-to-digest lignin, cellulose, and tannin, and some nitrogen as well. Fungi prefers this kind of material and have the enzymes to break it down. Only then can bacteria attack it. Other important factors. The bacteria in compost will tend to buffer pH around 7 to 7.5. Fungi in compost will tend to buffer the pH around 5.5 to 7. So you want some fungi in all your compost to prevent them from getting too alkaline. The more fungal material in your compost, the lower the pH to a point. Inorganic fertilizers, pesticides, herbicides, miticides, and fungicides kill off soil food web members, and therefore have no role in composting. Materials that go into compost piles should be free of these chemicals. Chances are they will break down over time, but maybe not before the compost is spread. And why take risks with chemicals when you don't have to? In addition, since many of these chemicals are non-selective when it comes to microbes, they can interfere with the composting process itself by eliminating microbes that contribute to the heat and decay. The size of the material put into a compost pile is also important. Too much fine particulate matter, and the pile will compress and quickly go anaerobic. If the material is too big, there will be so much air diffusing through it that the pile will heat up too much. If the material is too large, it won't decompose properly or fast enough because the bacteria can't get into it quick enough to establish sufficient populations. Hey, Moody. There is a fine balance when it comes to size of materials put into a compost pile, and only experimentation will give you the understanding you need and finally the control you want. Next, a compost pile requires a minimum amount of mass, approximately 3.5 square or round, 1 cubic meter, in order for it to heat properly. You can make your piles bigger, but the increase in size creates more work, as the entire pile has to be aerated or turned at least a few times to keep it from going anaerobic. In our experience, a 6-foot pile, wide and tall, is about as good as you will want without a lot of mechanical help turning and aerating it. It is quite easy to make compost literally in a pile, dumping ingredients right on the ground and mixing them. Some prefer a caged area to contain the material and to make the turning easier. A single ring of fencing or chicken wire, 3 feet in diameter and 4 or 5 feet high works great. Ooh, Jake. Good morning.
What you up to, buddy? Oh, you having a good day? Oh, yes. Sergeant. I love you.
Oh, I lost my space. Using a wooden pallet or screen supported on concrete blocks at the bottom of the pile will allow air to circulate into the pile, making it much less work to maintain. Some composters swear by revolving bins for making compost. In go the organic materials, and to aerate, all you do is spin the drum a few times. Once you figure out how to keep the materials in the bin from getting too moist, a chronic problem with enclosed systems, these can be very effective. Again, you will need to experiment to suit your tastes and needs. Whatever your setup, you will need to keep an eye on the pile's moisture. Place materials in layers of 4 to 6 inches, 10 to 15 centimeters, alternating between green and brown, and make sure each is moist. Once metabolic activity has started, you will need to make sure that the pile stays moist for the entire composting process. Since you don't want the pile to be wet, this encourages anaerobic activity. Make sure to mix wet material with dry material if necessary. If you are composting in a dry climate, flatten out or make a concave impression in the top of your pile to collect what rain does fall. Similarly, if you are composting where it rains a lot, cover the pile with a tarp or consider making compost in an enclosed bin. If a pile is too moist, it won't heat properly. You should be able to take a handful from your pile and squeeze just a few drops of water from it, but no more. If your pile does get too wet, then add dry materials or turn the pile. This is hard work, so it is better to get it right in the beginning. Hot composting will kill weed seeds and pathogens in most cases, but there is no reason to risk adding diseased material or really noxious weed plant material to your pile until you get the hang of the process and can distinguish composting from what we can tell is merely almost compost. There is a big difference. You have to finish the composting process to ensure the pathogens and weed seeds have been destroyed. How do you know you have good compost? Test it. You can send compost out to a biological testing lab, but an easier and cheaper home test is to smell the finished product. If it smells bad, like vomit, or putrefying matter, or vinegar, then it contains anaerobic organisms and their byproducts, and should not be used. If it smells like ammonia, then it is not finished. In either case, aerate it to change these conditions, and let it sit for a few days before you give it another nose test. You know what fresh soil should smell like? Good compost should smell clean as well. You could also plant something in it. Good compost supports plant growth. If there are not enough predators eating the fungi and bacteria, then the nutrients they hold won't be cycled, and you will be able to tell by the plant deficiencies.
Oh.
Compost for the lazy. A modern mix for instant compost requires 3 cubic yards of brown tree leaves and a 50-pound bag of alfalfa meal from an animal feed store. This mix works even better if the leaves are shredded so the bacterial microbes can get right to work decaying it. If you don't have access to alfalfa meal, start with equal volumes of grass clippings and leaves and work from there. If this pile heats up too much, use less grass. If it doesn't heat up enough, use more grass. This assumes moisture and air are adequate. We learn from experience that if you spread fresh grass clippings out and let them dry for a day or two before adding them to a compost pile, they won't mat or smell. Make your pile in layers, starting with 4 inches, 10 centimeters of leaves, followed by a layer of alfalfa meal of the same thickness, another layer of leaves, and another layer of meal, and so forth. Water each layer lightly and then add the next. Add sticks and branches as you go along to increase air circulation through and to the middle of the pile. Once you have accumulated at least the 3 cubic yards of organic material Once you have accumulated at least the 3 cubic yards of organic material needed by your army of microbes and other soil food web organisms, they will go to work. Heat will be noticeable in 24 hours. Thereafter, you need to monitor the temperature. It shouldn't get over 150 Fahrenheit, 65 Celsius, or cool down much below 104 degrees Fahrenheit, 40 Celsius. Turning the pile will increase the heat until the pile reaches the mature stage, after which it won't heat up when you turn it. Turning lowers the temperature temporarily until the microbes start working. I'll have to read that. It's out.
Mm-hmm. Moody, where's your treatment?
Oh boy, where was I? Once you have accumulated at least the 3 cubic yards of organic material needed by your army of microbes and other soil food web organisms, they will go to work. Heat will be noticeable in 24 hours. Thereafter, you will need to monitor the temperature. It shouldn't get over 150 Fahrenheit, 65 Celsius, or cool down much below 104 Fahrenheit, 40 Celsius. Turning the pile will increase the heat until the pile reaches the mature stage, after which it won't heat up when you turn it. Turning lowers temperatures temporarily until the microbes start working in concert again. Again, water will cool down a pile. If this sounds like too much work for you, try cool or cold composting. Simply pile organic matter in a corner of the yard and leave it. This material will eventually decay, only very slowly. Cool composting can take a year or more, versus a few weeks or months for hot composting. The end result is compost, however, and as long as it contains the proper set of organisms, it doesn't matter which system you use. Note that worms, beetles, millipedes, and other micro and macroarthropods will be represented in higher populations in cool compost. It is therefore a good idea to keep a cool compost pile going at all times. No matter how energetic you are, the diversity of soil organisms it adds can only help your garden. In the soil food web, higher member diversity means a better ability to eliminate pathogens or control them, either by direct attack or by competition for nutrients and space. Vermicompost. Processing organic materials through earthworms make vermicompost, which is almost always bacterially dominated. Few, if any, fungi are involved in worm digestion. Heat is not involved, as this would kill the worms. Instead, the worms, that is the bacteria inside them, digest the materials and create castings. You can buy special earthworms for this job, and buy or make a small bin to keep them in. This can be a simple wooden or plastic box, just out of the bin. Vermicompost has a bacterial dominance. The castings, coated with a polysaccharide as well as carbohydrates and simply proteins, are perfect for supporting good bacterial populations. Good starting materials for vermicompost include food waste, no fats or meats, paper, cardboard, leaves, and green grass, or you can use the same materials as you would to start a normal compost pile. If your material contains weeds, thermally compost them first before adding them to the bin. This prevents unwanted seedlings from growing in the worm bin. This prevents unwanted seedlings from growing in the worm bin. Any brown materials need to be shredded or otherwise broken up, so the worms can ingest it quicker. With luck, your materials will also include some microarthropods to help physically break down the material for the worms. Putting your bin outdoors will encourage arthropod and insect activity in it. Inoculate your soils. It doesn't take much compost to impart life to the soils. To inoculate your soils, put a 1/4 inch to 1 inch, 0.5 to 2.5 centimeters of the appropriate compost, fungal, bacterial, and balanced, around your plants. Fungal compost should be applied around trees and shrubs and most perennials. Bacterial compost is most appreciated in veggie and flower gardens and lawns. Review soil food web gardening rules 1 through 4. Compost can work its magic in the soil in as little as 6 months. After only that short period of time, new soil life will be evident in the first 6 to 15 inches, 15 to 38 centimeters of soil inoculated. With this new life comes all the benefits of soil food web. Decompaction, aeration, better water retention and drainage, and increased retention and availability of the nutrients. After a year, the soil life will be down as deep as approximately 18 inches, 46 centimeters. Gathering the materials and making a compost pile does take a certain amount of work. The benefits derived from compost, however, are almost incalculable when it comes to managing the soil food webs in your life. Compost is an indispensable soil food web gardening tool. And that was chapter 16 of Teaming with Microbes, The Organic Gardener's Guide to the Soil Food Web, Revised Edition, by Jeff Lowenfels and Wayne Lewis. Do the microbes produce a gas byproduct? Yes. Actually, it was described in the book, the anaerobic bacteria, the bacteria that, what was it? Archaea anaerobic bacteria.